This tutorial video will review how to submit your custom vector to Twist. Currently, you will need to submit your custom vectors using this custom vector submission form, which your account manager or Twist's customer support will send you. Twist does not use traditional restriction enzyme-based cloning. Twist uses a proprietary method of assembly which does not require you to add any additional flanking sequence, including restriction sites, to your inserts. So please do not add any extra nucleotides to your inserts in anticipation of restriction enzyme-based cloning. However, if you plan to use restriction enzyme-based cloning downstream, then there are two options. In option 1, if the restriction sites are not present in your custom vector, then ensure to submit your insert sequences with the whole restriction sites flanking your inserts. Otherwise, they will be completely absent in your final product. For option 2, if the restriction sites are endogenous to your custom vector, then ensure to include the whole sequence of the restriction sites when pasting your vector sequence into the form. Do not include these restriction sites on your insert sequences as well, otherwise they will be synthesized and thus duplicated on your final product. After filling in your contact information, enter the vector details into this submission form. The top row of the table includes an example demonstrating best practice for filling in the form, whilst the 3 rows below contain examples of how not to fill in the form. In column A, the custom vector ID is simply your unique name for the vector. In column B, the insertion site ID, this is just your unique name for the insertion site in the vector. In column C, you need to copy and paste in the 5 to 3' direction the exact sequence of the custom vector DNA that you will be shipping to Twist. So, in my designer software, I will copy the whole in silico vector sequence and then paste it into column C. In column D, copy and paste the vector plus an example insert sequence, ensuring that they have the same directionality. This will be an example of a final expected construct after the cloning. I would like to insert my insert sequence in the multiple cloning site in between BAM-H1 and ECHO-R1 restriction sites, which I want to use for my own experiments further downstream. So note that this is like option 2 that I mentioned earlier, where I have endogenous restriction sites in my vector. I have created an in silico representation of this, where I have my backbone vector sequence plus the example insert at this specific insertion site. I will copy and paste this whole construct into column D. It's important to note that only one example insert sequence is required per vector, just to demonstrate where each insert sequence should appear in the vector. In column E, copy and paste the insert sequence without any extra bases. So, back in the designer software, I will copy just my example insert sequence. Notice that I haven't included any flanking restriction sites on my selection because these are already present in my vector and I don't want to duplicate them in my final construct. After pasting all the sequences, column F verifies the presence of the insert, which you just pasted in column E, at your desired location in the vector in column D. By default, the formula is set to insert found, so only if the insert is not identified will you see insert not found. The order cannot be processed until column F reads insert found. Column G detects the presence of any non-ATGC bases in column C, D, and E. Only ATGC bases can be accepted. Please then enter the resistance marker, and if known, the copy number and the origin of replication. For adding subsequent vectors, please add these in new rows below. Likewise, if you require multiple insertion points for the same vector, then these will each need to be entered as a new row because each new insertion point requires a new vector onboarding strategy. For shipments, please send at least 50 microliters of plasmid DNA at concentration of at least 100 nanograms per microliter and send to this address. And please provide your account manager with your vector shipments tracking information. Once Twist have received the vector, and provided you with an onboarding start date, it should take 10 business days to onboard after the provided onboarding start date. You will then see it appear in your e-commerce account in the Jeans app under the Vector drop-down menu. You can then select the relevant insertion points when placing your order of clonal genes. And you can also do this for multiple genes simultaneously too. Feel free to contact us for any further inquiries using our live chat, or you can email us to customersupport@twistbioscience.com.